There's nothing as full of potential as a newborn child, futures yet to unfold. Yet for so many Indian children, especially those from low-income homes, this is the time that proves most perilous. They must dodge an army of bacterial and viral infections. They must fight off diseases that seem routine for most adults, but can be fatal for them, diseases like diarrhea. But while many children succumb to diarrhea, there are some whose immune systems fend off the attack and in fact develop powers to resist them all lifelong. Can this natural immunity found in some be harnessed to save all? That's a question of science. For developing nations like India, there's nothing more dire than the problem of infant mortality. Too many children fall prey to diseases that are simple enough to cure. One such disease is diarrhea that claims 2 to 3 lakh infants every year. Diarrhea is a debilitating condition that causes severe gastroenteritis and dehydration. Its most common cause, the rotavirus. Rotavirus is an extremely contagious virus that's found commonly in our environment. It spreads through fecal matter or through food and water. Once it enters the body, it attacks the cells of the small intestines, leading to acute gastric problems. If treated properly and immediately, it not only resolves but also ensures that the body is immune to it for the remainder of its lives. You could well ask, if children can be immunized against diseases like polio, measles and smallpox, why can't there be similar preventions for rotavirus diarrhea? The answer is, there can be. This may look like a normal clinic with young parents bringing their infants for routine checkups. But there's nothing routine about these proceedings. Because this is one of the sites for an exciting drug trial that could save lakhs of Indian children with a single dose. Each child here is contributing to bringing India closer to having its very first completely indigenous rotavirus vaccine, also known as the Rotavac. The Rotavac has taken over 2 decades to reach this phase. And it all began with a pediatrician going about his daily job at the AIIMS, or the All India Institute of Medical Sciences. In the 1980s, Dr. M.K. Bhan was an associate professor at AIIMS. It was here, while treating the stream of babies that passed through the hospital, that he made a startling observation.
I was actually testing a diagnostic kit for rotavirus. And so I went to the nursery and I took stools of all the babies. When we ran this test, we call it a ELISA test, when we ran it in these stools, we were quite surprised to find that almost all the babies were positive. They were all getting infected with an unusual type of rotavirus, but they were not getting sick. So it created a curiosity in us whether this virus strain is changed or altered in its gene structure or function in such a way that it doesn't cause disease, but it causes infection and therefore it will cause protection. So is it a natural vaccine?
This led to the next logical question. If we could study the root cause of this immunity, could we use it to immunize other babies as well? In other words, had Dr. Bhan stumbled upon a natural vaccine? Dr. Bhan's finding was an echo of another doctor's. This doctor had lived half a world away and over 3 centuries in the past. In the 1760s, Dr. Edward Jenner of England made an observation. He noticed that farmers who reared cattle seldom succumbed to the dreaded smallpox. On further inquiry, he learned that most of them had suffered a milder form of the disease in their childhood called cowpox. Childhood cowpox had somehow rendered them immune to smallpox. Dr. Jenner went one step further. In 1796, he inoculated an 8-year-old boy with cowpox. 6 weeks later, he exposed the boy to smallpox. To his joy, the child had developed an immunity. How had this occurred? The child's body had definitely been infected with cowpox virus, which was similar to but less harmful than smallpox. The infection was enough to trigger the body's immune response. Immediately, it began producing antibodies to combat the virus. So, by the time Dr. Jenner exposed the child to smallpox 6 weeks later, his body had developed brand new weapons to fight it off. Thus, the world was given a new vaccine, or a biological formula that used a small amount of disease-causing agent to build the body's defenses against the disease itself. Back at AIIMS, Dr. Bhan had made Jenner-like observations with rotavirus, and he wondered what was the next logical step. Where would the chance finding lead?
With most chance findings, the important thing is whether you follow them or not. And in this case, we were lucky that we actually decided to follow it up, and our curiosity got the better of our lethargy.
Dr. Bhan was not the only scientist who was looking to crack the formula for a rotavirus vaccine, or Rotavac. The American Dr. Roger Glass had been studying similar strains of rotavirus across 4 continents. One was a doctor from a country that had never made a vaccine from scratch. The other hailed from a nation that had the expertise to do exactly that. Both sought to defeat the same disease, Rotavirus Diarrhoea. Fortunately for them, it was around this time that India and the US of A jointly launched the Indo-US Vaccine Action Program, or VAP. A determined effort spearheaded by the Department of Biotechnology, Government of India, and the National Institutes of Health in the US.
Especially the below 5-year-old children, where we need to vaccinate them some 6 or 7 vaccine preventable diseases. So that was idea in 1987-88 under Indo-US collaborative projects by involving a leading Indian and US scientists to develop the new and improved vaccines. So basic idea was to really produce a vaccines which affordable cost for Indian context into that.
With the backing of not one but 2 government agencies, it was time to unravel the mysteries of the unknown rotavirus strain that was discovered at AIIMS. So far, scientists knew of only 5 to 6 major strains or types of rotavirus, of which strain A was the most commonly understood. The AIIMS strain would have to be sent to virology experts abroad for further studies. Extensive genetic studies were carried out at the Center for Disease Control and Prevention, or CDC, in America. Here they made yet another amazing discovery. At the molecular level, the strain had a strange mutation. Besides carrying human proteins in its genes, it had one genetic segment that seemed to be derived from bovine. In other words, cattle like cows and buffaloes. It was this bovine mutation that, much like Dr. Edward Jenner's cowpox virus, seemed to give humans the power to resist disease.
This virus, which we call 116E, had one gene of animal origin, bovine origin, and 10 genes of human rotavirus origin. And it was our impression that this one gene of animal origin is the reason why babies don't get infected with this. This is how genetic evolution of viruses occurs. But it can also happen in the human gut. Imagine a child who is infected with 2 different viruses. So they co-cultivate and transfer genes to each other, and then when that child sheds the virus in the stool, a new virus is born.
The AIM strain of rotavirus was renamed 116E, and back home in India, further studies were carried out to study how it behaved. All these studies made one thing eminently clear. Neither the rotavirus nor its disease were the same across the world. For example, India had a far more diverse collection of rotavirus strains than in any other part of the world. While the disease was seasonal in Europe and the US, for India it was year-round. Most significantly, while nearly 2 lakh children die of rotavirus diarrhea every year, less than 10 die in the United States. In these circumstances, could India rely on research done in the West alone? Wasn't it critically important to start a specific vaccine development program targeted towards India?
Like any other organisms, like microorganism, there will be some specific to Indian context. Rota, this neonatal diarrheal strain isolated by Dr. Bhan, we consider it specific to Indian children. That's the reason why I say India specific rotavirus vaccine. That's why we gave the name of 116E, and from there we started characterization of the strain and working on that, adoption to the vero cells. All this process is gone through in a very detailed manner for 10-15 years into that.
The Indo-US Vaccine Action Program supported research on 116E throughout the early 1990s. But they weren't the only ones on the chase for the Rotavac. Dr. Durga Rao of Bangalore and Dr. Harry Greenberg of Stanford University, USA, had been working on another strain of rotavirus called I321. Like the 116E strain, it had a mix of human and bovine genetic segments. And just like 116E, it showed promise of being a natural vaccine against rotavirus diarrhea. But there was one fundamental difference.
They had a strain which was essentially an animal rotavirus, but there was one human gene in it. We had a strain which was essentially human but had one animal gene. So the question was, which one would give the best balance between safety and efficacy?
There was only one way to answer this question: put both candidates through rigorous tests. For that, India would have to produce the vaccines first.
Then 98-99, we invited the public sector company or private sector company like Cadila, and Bharat Biotech has showed interest into that. Team was created and we visited those sites. Finally, we selected Bharat Biotech because Dr. Ella Krishna was very keen to develop this vaccine along with our entire group. He also thought this consortia, this group is excellent in the international level of scientific merit.
BBIL may never have made a vaccine from scratch before, but what it lacked in experience, it made up for in passion to succeed. It opened its doors to American experts from the CDC. The CDC had already helped in genetic and virology studies of 116E and I321. Now it stepped up to help the Indian biotech company set up state-of-the-art systems to manufacture vaccines.
I had a passion. Only one thing is that India never developed a new vaccine. I thought it's a time that, being a new generation entrepreneurs, that we should develop some new vaccines and India should stand on its feet. Like making a cell bank was challenging. We don't have any US FDA approved cell line in this country. And we were the first one to develop US FDA approved thanks to international collaboration.
As pilot batches of vaccine candidates emerged from manufacturing plants on both sides of the Atlantic, it was time to start testing them. And so, Phase 1 began in the early 1990s with testing the vaccine not on babies but on adults.
Adults are given the vaccine, some are given the vaccine, some are not, and you look at what events in terms of, let us say, fever or diarrhea or vomiting, skin rash, things like that did they have. So we did a study in adults, then we did it in 2 to 12-year-old children, and then we came to the target age group, which was infants.
The question now loomed, which strain would emerge stronger, 116E or I321? With each test, the answer became clearer. It was 116E that showed a better immune response. The Rotavac brought together diverse research teams from different institutes across India and the US. And it partnered government funding agencies with global funding ones like PATH and the Gates Foundation. They all had a singular motivation: social innovation.
To solve big problems of this world, you need team science, and you need innovation through large communities of institutions and people. Now, how do you make that real? Our natural urge is to compete. How do you substitute competition by cooperation? This is a great 21st century challenge.
Through the 2000s, the Rotavac went through a series of development stages, each stage accompanied by stringent tests. After judging the vaccine to be safe, it entered the next phase.
In the phase 3, there were 3 sites, and at each site there were a minimum of 150 staff. So when you have a team of 500 people and you want to, at different categories, everybody needs to follow the same methods. You need to standardize people. There are periodic trainings, retrainings, monitoring, audits. So that's the challenge.
March 2011. After over 2 decades of R&D, the Rotavac was ready to enter the most critical phase in its march towards completion. Phase 3 of the clinical trials, where it would be tested on babies to see how safe and efficient it was. It was a first for India, which had never before tested a vaccine manufactured in the country.
We are going to look at groups of children who would receive the Rotavac and the other group of children who would not receive the Rotavac. And then when we look at it at the end of the study, we hope to see that the children who have received the Rotavac would have lesser severe rotavirus gastroenteritis.
Nothing on this scale or standard had ever been attempted in India for a brand new vaccine. The tests would not only have to adhere to Indian regulations, but would also have to live up to the global and gold standards set by the US Food and Drug Administration or FDA. The trials involved 3 sites across India: Delhi, Pune and Vellore. Approximately 6,800 babies were selected as subjects and given 3 doses at the ages of 6 to 7 weeks, 10 weeks and 14 weeks. The subjects were closely monitored and followed up on for 2 years.
We have this huge, like an army of team and every team has different responsibilities, and so to get all of them together and ensure that all of them are in sync with each other, and also to ensure that everybody understands they are a piece of a bigger team, not, that they cannot do things by themselves. That is what needs to be created.
The process: selected babies come in. Their families are taken into private enclosures and asked for informed consent. There is a thorough medical checkup of the baby. They are given a special ID number. From now on, this and not their name, family or house number will be their identifier.
We want to be very sure that we are looking at the efficacy that we are seeing is collected in a way that is not biased. By that I mean, if I know that a child has received Rotavac, I may tend to look at that, pay more attention to that baby and not pay more attention to the baby who has received the placebo.
Then they are ready for the Rotavac. But not every child receives the same vaccine. These are double-blind, placebo-controlled tests. It means that if 2 babies receive the 116E Rotavac, 1 baby receives a non-Rotavac but harmless solution. None of the families, investigators, lab staff or on-call pediatric doctors know which baby gets which dose.
After the Rotavac is given, is an oral vaccine. So after that, we give them the childhood vaccines. After they are given the childhood vaccines, each and every baby gets a tag. We had made a tag for them, and the subject ID is written in the tag, as well as the time when the last vaccine was given to the child. And the reason being, as part of the safety for all children and as mandated by the government.
Over the next 2 years, the infants would be closely monitored to see which ones developed severe gastroenteritis and which ones didn't. At regular intervals in the follow-up, samples collected were routinely analyzed at THSTI labs. The success of the Rotavac hinged on these results.
These results are comparable or very similar to the results from other rotavirus vaccines in the developing world.
Today, not just is the Rotavac a reality, but its next-gen product is being tested. In the process, Indian science has grown. We have learned how to do team science. Our industry has learned new technologies. And we have gained distinction in clinical trials. But none of them are as significant as the final achievement which the developers of Rotavac eagerly await: inclusion in the National Immunization Program of India.
So this is a wonderful model which we established. Probably we will take this as an example and move forward other vaccines which are in the pipeline.
Discovery cannot be learned by textbooks. It has to be learned by experience. So you plunge and take a chance, take a risk that you might fail, and you learn.
If you'd like to share your feedback on today's program, please send your suggestions and comments to Vigyan Prasar, C24, Qutub Institutional Area, New Delhi 110016, or you can mail us at info@vigyanprasar.gov.in.